So we wind up having extra copies of specific genes on the B chromosomes. And a lot of these genes are aberrant in canine cancer. Also, some of these probes were identified in a complementary study identifying a sequence that is found on the red fox chromosomes. As I talked about before, we did array painting experiments. So the red fox chromosomes were flow sorted, and we wound up with a sample of the B chromosomes from the red fox, and this was labeled and then applied to microarrays. And so what we were able to find is specific sequence present on these chromosomes. And this is at a much higher resolution than the specific single locus probes that I used. So to kind of guide you through this data, this is a red fox chromosome, or red fox metaphase chromosome spread using a probe that represents the c-KIT proto-oncogene. What probably sounds familiar to everyone is that c-KIT is involved in a lot of different cancers. So this red probe represents that sequence, and you can see that on red fox 2 here and here, that there is a red signal. And that's what represents the region of c-KIT that is found on dog chromosomes, is found on all of the different canids. In addition, there is a huge signal on the B chromosomes. So here, I kind of laid out all of the B chromosomes so you could get an idea of the range of signal. What should be very obvious to you is that the signal is very much larger and more intense. And so we believe that this represents a lot of tandem duplications of the sequence. When examining the Chinese raccoon dog, and remember these are the larger B chromosomes, we see specific patterns. And so it's a way of identifying B chromosome 1, 2, and 3. And this is an ideogram to give you a more simplified idea of where these probes lie on the B chromosomes. When we examined the Japanese raccoon dog, we had a simpler story, or simpler data, showing that this probe lies in a subtelomeric region. However, on the third B chromosome is a little more proximal. So as a whole, we wind up with very large and intense sequence on the red fox B chromosome, and sequence in multiple regions on the Chinese raccoon dog. However, kind of a more simplified region in the Japanese raccoon dog.